I'd like to begin today's proceedings by acknowledging the traditional owners and the custodianship of the lands on which we meet. We pay our respects to their ancestors and their descendants who continue cultural and spiritual connections to country. We recognise their valuable contributions to Australian and global society. Good afternoon, everyone. For those of you that don't know me, my name is Brett Nixon, and it gives me tremendous pleasure to welcome you this afternoon to this very special occasion. Again, for those that don't know me, I'm the president of the Society for Reproductive Biology here in Australia. And again, in this capacity, I'm very, very pleased to be here today. We're here to pay tribute to two incredible scientists who have left an indelible mark on our field of reproductive biology. So we're getting a bit of feedback. We're right? Okay. Before launching into recognising those scientists, however, I'd like to take a moment to acknowledge and extend a very warm thank you to the sponsors of today's event. This is being co-hosted by the Society for Reproductive Biology, our society here in Australia and New Zealand, as well as the Society for the Study of Reproduction in the USA and the Society for Reproduction and Fertility in the UK as well. I'd also like to make special mention of the Faculty of Medicine at the University of Queensland for their sponsorship of this award, or for this venue rather, and making this beautiful venue available to us today. We have Karen here representing UQ as the Associate Dean of Research. And if anyone doesn't know Karen, she's also an exceptional reproductive scientist as well. So we're grateful to have you along, and we're very grateful for your support as well. Whilst I'm thanking people, it'd be very remiss if I didn't extend a very sincere thanks to Cassie, who has really gone above and beyond to put all this together. The venue, the catering, just all the coordination that's gone behind this. It's all been really Cassie that's done it, and I'm very grateful for your support. Thanks very much, Cassie. Okay, so the first of the scientists that we're here to recognise today is the late Professor Roger Short, who sadly passed away on the 6th of August 2021. By all accounts, Roger was a true luminary in our field, a mentor to many, an eloquent speaker, and an incredibly creative thinker. In the time I have here, I simply don't have the possibility of capturing all the impact that Roger had within our field. But suffice to say that his work has helped to improve the lives of many people in many countries and in many disciplines that sit well beyond our sphere of reproductive biology. We're incredibly fortunate to have his wife, Marilyn Renfrey, here today, and his close friend, Jeff Shaw, who have put together a presentation to celebrate both Roger's career and his life. And immediately after the oration, we'll play that for the audience as well. Among the many features of Roger's career that struck me were his fascination with just about everything, I think. He really had this sense of trying to understand how everything worked around him. He had an unparalleled imagination, and he had an endearing quality of making young researchers feel important and valued in terms of sharing their research interests as well. And it's in that spirit that the sister societies of the SRB, the SSR, and the SRF have tried to honour Roger and his career and his life through the establishment of a highly prestigious award that is to be bestowed annually on an exceptional young scientist who has not only made a significant contribution to our field of reproductive biology, but also epitomises Roger's spirit of discovery and excitement about the way he did his research as well. We had a very competitive field of applicants for the inaugural award, and the selection committee of SSR, SRF, and SRB are delighted to announce that the inaugural recipient of the R.V. Short Medal in Oration is Professor Niamh Ford from the University of Leeds. We're incredibly fortunate that Niamh has made the journey all the way out to Australia to give this oration, and we're very grateful for that you've come, Niamh. For those that haven't had the pleasure of meeting Niamh at this stage, or have rather had the pleasure of meeting Niamh, you'll be aware that she is an absolutely delightful person, an incredible scientist, but also very humble as well. And I can see her dying as I'm starting to talk about her career. Yeah, I'm sorry, Niamh, I know this is the hardest part of the whole thing. And again, I really don't have time here to pay tribute to everything that Niamh's achieved over the course of her career. She is on a phenomenal career trajectory, and I just want to roll off a few highlights of her career to date. Niamh is a professor and chair of molecular reproductive biosciences based at the School of Medicine at the University of Leeds, where she's also a co-founder and co-director of the Leeds Omics Research Institute. As we'll hear today, Niamh's research program primarily focused on understanding the molecular interactions that occur between the endometrium and that of the embryo that are required for successful early pregnancy. And a defining feature of Niamh's research is that she doesn't just study a single species, she studies this across a broad range of mammalian species and is really interested in the comparative aspects and what they can tell us about the reproductive process. Her research interests also extend to how the maternal environment and the sex of the embryo influences these types of interactions as well. And to answer these types of questions, Niamh and her team use some really quite cool technologies, not only in vivo technologies, but also in vitro technologies such as microfluidics, organoids, extracellular scaffolds, and very sophisticated and integrated omics technologies as well. And this work is bringing us ever closer to a molecular understanding of how both protein coding and non-coding regions of our genome regulate endometrial function, with implications that extend to improving food security, improving fertility in our species, and also improving overall health of our species as well. Niamh is also a proven leader in facilitating collaborative interdisciplinary research and is absolutely committed to enabling the development and growth of the next generation of scientists as well as being a very strong advocate for our field to help realise the societal impact of our fundamental research, not only her own, but that of the broader field as well. I could go on, but again, I think Niamh might kill me if I do. She's really come a long way, and we're very grateful to have you here, Niamh, to be presenting this inaugural lecture. You're an incredibly deserving recipient of the inaugural RV Short Medal and Oration, and I'll now hand the floor over to you. Thank you.
Thanks very much for that introduction, Brett. I really, actually, before I start, I really want to thank the SRB for hosting this lecture. It really is an absolute privilege to be here today to speak to you all. And a special shout out to Cassie as well for helping organising this. I want to start off by saying this is a really strange type of lecture to give. So, you know, as scientists, we're really used to being told you've 30 minutes to talk about your science, and you know you can do that for probably longer than you should, and you know exactly how that goes. Whereas for this lecture, I thought it was I spoke to lots of people about what the lecture might look like or sound like or entail, and there was a general scope of anything or whatever you would like to talk about, Niamh, which sent me into a slight panic, I don't mind saying. However, when I quelled the panic a little bit, I started to think a lot about what this type of lecture could be or should be, informed obviously by Roger and kind of his approach to things. But also I thought it might be helpful and useful for more earlier career researchers as well to understand how I'm standing here today to talk to you all. So that's kind of the theme that I've managed to thread through the talk today. And it kept coming back to environments. So the environments that I have been exposed to, the uterine environment, which is a really important component of the science I do, and broader environments. And that's kind of where I came up with the title, Why Environment Matters, Understanding Life, the Uterus and Everything. And so because this is a slightly different approach to speaking, I'm going to give you an introduction as to what I'm going to speak about today. Hopefully, when I manage to get the thing. Yes, hello. So I'm going to talk to you about 3 different types of environment: my past environment, the present environment, and sort of a little bit about the future, where I think our field is going to be really important going forward. And as I talk through the past, the present, and the future, I'm going to tell you a little bit about me. I'm Niamh, hello, not spelt like it's pronounced. I'm going to talk an awful lot about the uterus, which obviously is the science that I'm really, really passionate about, and why it's so fundamentally important to understand this really, really fascinating organ. It's the only organ that supports growth and development of all other body systems, and so it's really, really critical that we understand this. And then I'll touch a little bit on some of what I think are the major future challenges in terms of environment as well. So if I talk about the environment in the past, I want to introduce you a little bit about me and who I am and where I've come from. So I grew up on the west coast of Ireland, in a place called Galway. It's a beautiful place, slightly disingenuous with the pictures because there's sunshine in them. It rains almost all of the time. But I grew up in Ireland in the 1980s, and I'm the youngest of three. So I have an elder brother, John, an elder sister, Jean, who's here in the audience today, who did try and kill me when I was relatively young. So maybe have a word with her about that later on today. But I was the youngest of three kids. And growing up in Ireland in the 1980s, everybody was poor, so no one was rich. Going to university didn't seem like a thing most people did. However, the environment I was raised in was a really positive one, because even though neither of my parents had finished the equivalent of high school or secondary school, my mom in particular was really vociferous about reading and also about education. And I think growing up in that environment where, okay, so the generation ahead of us hadn't managed to be able to access third level education, but there was no barrier to us being able to access it, right? So we were always really, really encouraged to read a lot and also do as much study as possible. And obviously I've taken that to the extreme because I still haven't yet left a university. But I think that was a really important environmental component as well. I think the other thing is that even at an early age, I was showing sort of an unorthodox approach to problem solving. So this demonstrates my unusual approach to addressing how to get up a slide, which I think is infused a little bit in the science I do. But it's also demonstrating another key component, I think, for being an academic, which is a bit of stubbornness in a person. And so I was really fortunate to have this type of environment. And then another key important environmental component was in the early 1990s, the Irish government actually removed the barriers to second or third level education by removing fee payment. So you could actually go to university for free. So the environment I was in was very supportive family, very education focused. Now there's no financial barrier. And so you can go to university as a thing. And I did, but instead of going to the university, 10-minute walk away from my parents' house, I decided age 16, sure, I'll go to Dublin instead. So I went and did my undergraduate at Trinity College, where I studied biochemistry. And it was only when I was in the final year of my undergrad that I really got interested in reproductive biology. It wasn't actually taught that much in school or in first year and second year, but I did my equivalent of the honours programme, trying to isolate unsuccessfully an epididymal glycoprotein from a mouse. And that really sparked my interest in reproductive biology. And I thought, God, this is a really, really cool field. And so after I finished my undergrad, I then went on to work in a hospital lab for a little while to see if I actually wanted to do a PhD. And I discovered I was really bored. So I did apply and I got a PhD based at University College Dublin. And so this is where kind of my area of reproductive biology was really sparked. And actually, where I first met Graham Martin, he came to visit my supervisor's lab, Alex Evans, at the time. And my entire PhD was on the genomics of follicle development. And this PhD environment was really important, actually, for two reasons. One was because it was around the same time that omics became a thing, and that's a really big theme that's been woven through what I do and what my lab does now. And it kind of, my PhD journey started when omics were really coming to the forefront. The other thing was that my supervisor was quite a supportive supervisor. I used to pester him a lot for different opportunities. And so he gave me like lots of different opportunities to get training elsewhere. It was a well-funded group. And I think these are all really important environmental components that had me sort of contributing to how I'm here today. I did have some like slightly not delightful things in my PhD. And for those earlier career researchers, I finished my PhD with 0 publications. So again, this isn't the worst thing to happen to you. You will get there eventually. So after I finished my PhD, I begun what I think has been my favorite pieces of science that I have done so far in my life. And this is beginning to look at uterine biology. And so before I started my PhD, or after my PhD, I didn't know much about the uterus. And this was really, my postdoc was the start of my journey onto uterine biology. And just to set into context super briefly, we know that in like lots of mammalian species, some of these early pregnancy events can be easily recapitulated in the lab. There's a lot of conservation in terms of morphological events that occur in early pregnancy. And we know we can recapitulate the early stages, at least in vitro, quite well. However, I was solely focused on using cow as a model system, and this was really interesting to me because unlike humans and mouse species, cattle conceptuses undergo this rapid prolonged peri-implantation period of development, where the outer cells of the embryo or the trophoblast rapidly elongate. And this produces the pregnancy recognition signal. And establishing uterine receptivity to implantation is entirely driven by the actions of the hormone progesterone. And so some of the key components or functions of the endometrium is what we tried to sort out and figure out when I was doing my postdoc at UCD. And so basically, one of the really important environments I was exposed to at UCD when I was doing my postdoc. So one of the really important environmental components for my postdoc was I worked with a professor called Jim Roach, who had incredibly exacting standards about science and how experimental design was carried out. And that was really, really key for me because I was able to understand and address some really fundamentally important questions for uterine biology in cattle. And so we set about doing a series of experiments where we tried to understand how progesterone affected the endometrium, how that had modified the uterine environment, and how that contributed to pregnancy success. And so a couple of the key questions we addressed. Yes, this is the other environment that I managed to get exposure to while I was a postdoc was to spend some time in the US in the labs of Fuller Baser and Tom Spencer, both at Texas A&M and at Washington State University. And for me, these were really formative experiences. So in Dublin, I was the only person working on the uterus. I went to Texas, first of all, and suddenly there were like tens and if not hundreds of people working on the same organ and all thought it was really, really cool like I did. And so it totally changed how I viewed science, how I did my science, and understanding how great it is to have a critical mass of people all excited and enthusiastic about the same thing. And so I think that environment was really, really formative in how I did and how I approached my science. The other really key thing about getting to spend some time in the US was that I managed to begin to build my network of scientists, which is a really important component to the environment that we work in. And if you're a socially awkward human like I am, sometimes networking can seem like a really difficult thing to do, and something that people who are way slicker than me, I would always think are really good at doing. But actually, if you're at a conference the chances are the other person is a bit awkward as well, but you have this really, really important, passionate part of your science that you have in common. And who doesn't love to talk about what they do, right? And so one of the really important things and the environments that really helped and has helped me throughout my career has been going to the SSR meetings. And I managed to build up this network. So I've managed to go to every single SSR meeting except for COVID. And it's really, really helped me and my career, particularly things around, so Karen Schindler, who does wonderful work for the Women in Reproductive Sciences, is really a strong advocate of nominating women scientists, and through these networks as well, people who've written me support letters that I'm really, really grateful for throughout, some of whom are in the audience. And it's really building that network and talking to people and interacting with them. And I think that's a really critical environmental component to the science that we do. Also, my sister pointed out I probably should have ironed these beforehand, but we'll gloss over that. Back to the science, right? So doing my postdoc, having an environment where very rigid scientific approach to things, expanding my network, spending some time in the US, really changing how I think about the science, enabled me to begin to answer some of these questions. So these are some of the questions we tried to answer when I was doing my postdoc, such as, if we increase progesterone in circulation, how does that affect embryo growth and development? Well, it has a substantial effect on that. And that is mediated indirectly by actions of progesterone on the uterus. This is pre-bio-render, don't judge me. I spent hours doing this in PowerPoint. But what we managed to find out was actually using an omics approach that progesterone acts on the endometrium and advances the normal temporal changes that do occur. And we were able to show that for the first time, that then results in getting advanced conceptus elongation, more pregnancy recognition signal, and then more likely that the pregnancy is going to be successful. We also managed to do the opposite, where we modified or suppressed progesterone from the corpus luteum, demonstrated we get delayed conceptus development and also delayed the normal temporal changes that occurred in the endometrium. The other key question we were interested in is understanding when the endometrium was able to sense the presence of this embryo or conceptus. And so using a microarray approach, does anyone remember them? We managed to show that actually it's on the day of pregnancy recognition, on day 16, that the transcriptomic response of the endometrium to the conceptus is observable. But we also know that some of these interactions are not just due to the pregnancy recognition signal. And this is one of the last experiments I did while I was at University College Dublin to try and identify some of these other conceptus-derived proteins. So you might be saying, great, Niamh, that's lovely. Why are you telling me all this information? Well, actually, while I was doing all of this kind of cool science, I was applying for jobs in Ireland unsuccessfully. There's not very many universities there. I never even got interviewed at the time. And so in around towards the end of 2014, I took the decision to move to the UK. So I applied for some positions, again, didn't even get interviewed for most of them, and finally got a faculty position at the University of Leeds. But bear in mind, this is the last experiment I did before I left Dublin, because I will come back to it in terms of science. So that brings me sort of to the present-ish. So when I moved to the University of Leeds, it was a really good university, but didn't have that critical mass of reproductive scientists. However, when I was recruited there, I was recruited on this 250 Great Minds scheme, which meant across the university, they were hiring 250 faculty to begin their own research groups. And so you can imagine how vibrant a community and a place that was going to be starting your own research group. And so it was really important for me, at least, when I was starting my own lab, that I was in this kind of vibrant environment of University of Leeds, lots of new people starting their research groups. And it allowed me to talk to and interact with lots of different faculty members that you wouldn't ordinarily get a chance to speak to. And I think that's been really key for what I've been able to do in my lab and the approaches that we take, because we do so in a very sort of interdisciplinary manner. And that's been sparked by speaking to people across different faculties. So that's been a really important environmental component to how I've been doing some science in the last few years. The other component is trying to build an environment that isn't necessarily there, but you can build one if you really try. So at the same time, when I started, a couple of other colleagues who use lots of omics approaches in their research started as well. And there was no omics community at the University of Leeds at that time. And so we decided, okay, we would quite like to have a community of people. There isn't one here, let's just do it ourselves. And so that started us establishing Leeds Omics, which is a virtual research institute, which I did with two of my colleagues and now great friends, Julie Aspton and Mary O'Connell, where we basically established a virtual research institute to help support, train, provide bioinformatics support, and really importantly for us with our fledgling research groups to develop that community and good environment and supportive environment as well for them. And so that's been a really, really important component of my research environment since I've been at the University of Leeds. I'm going to start talking about some of the science now in a minute, but I thought it was really important to say the science I'll talk about is the science that we've managed to get funding for. I think we all know we can struggle with getting grants and research funding, and I think it's really important to have a supportive environment for when that happens. So here is a screenshot of my rejections from the UK funding equivalent of NHMRC or the NIH. So as you can see, there's the odd successful littered amongst the graveyard of rejections, as I call this screenshot on this page. And I think it's really important when we know in this job that we would get professionally rejected like all of the time. It's really important to have an environment and people who will support you at the time when you have those rejections. And I just wanted to make a quick shout out to Lynn McEwen and Karen Forbes, who have been that for me at Leeds as well. So again, try and build your networks and have those supportive environments for you when you need them. So to the science, right? So when I was in Dublin, I worked exclusively on cattle. But the environment that I went to at Leeds was very interdisciplinary, and I got to talk to lots of different people. And so that allowed me to expand how I thought about science. What were the big questions that I actually really wanted to begin to try and understand? And so there was three main questions that we've been focusing on the lab at the moment. One is understanding this wonderful diversity of pregnancy events and pregnancy morphologies that we have in placental mammals. So why are closely related species, why do they have the same or similar type of implantation strategy, but a totally different placental phenotype? Why is that? What's regulating those and what's driving that diversity and difference? The second question that we're interested in understanding is why is pregnancy not very successful in different species? I think we've done really good work around understanding how the contributions of the oocyte, the sperm, the embryo, but I still think there's a lot of work around the contribution of the uterus to pregnancy loss that we really don't understand because it's such a difficult organ to study. So one of the questions again, what is driving pregnancy success or dysfunction in placental mammals? And then finally, understanding the developmental origins of health and disease. So we know that the uterine environment is reflective of the maternal and larger environments, and how do those stressors interact via the endometrium and the uterus to establish life course health trajectories. So start of my lab, talked to some people, and then decided let's look at these three really big questions. How are placental mammals diverse? Why does pregnancy loss occur? And what are the kind of uterine contributions to developmental origins of health and disease? And so I'm going to talk through a little bit some of the data we've generated for a little while. And I'll begin with speaking about how the embryo interacts with the uterus. So one of the really important questions that I've thought about for a long time is understanding this biosensor component of the endometrium. And so in the late 2000s, Eckhard Wolff's group in Munich and Olivier Sandra's group in France identified that if you transfer in embryos with different developmental competencies, the endometrium can sense those competencies and they respond differently. But no one knew what was mediating or what the mechanism was. So I happened to be in Brazil in like 2016, talking to some people who work on extracellular vesicles, and we thought, well, maybe this biosensor component might be mediated by extracellular vesicle cargo. So we set about designing this experiment where we transferred in high quality in vivo embryos, intermediate quality in vitro, and low quality cloned embryos. We recovered them on day 16, which is a key stage of pregnancy in cattle, recovered the endometrium, the conceptus conditioned EVs, and begun to understand what the differences in these cargoes were. And what we've identified so far is that we think that some of the endometrium does respond differently to these different types of embryos with different developmental competency. And we think this is mediated in part by different cargo loading into the extracellular vesicles. And these are modifying the endometrial transcriptome accordingly and modifying pathways that are key components of implantation success in these models. So we're beginning to understand a little bit about how the biosensor capability of the endometrium is mediated in part. We also begun to investigate some of these proteins that I'd identified when I was doing my postdoc at UCD, and we began to analyze and understand what types of these proteins are, how common they were among placental mammals, to try and understand a little bit more about not just the pregnancy recognition response of the endometrium, but actually the response to other proteins that we know the embryo is producing around implantation and what jobs they may be doing. And so one of my students has worked a lot on this protein PDI. She looked to see how similar the sequence of that protein is amongst placental mammals. It's super homologous. And the notion is that if a protein is really highly conserved in terms of its amino acid composition, it potentially could be doing the same job. So we asked the question, if this protein is very similar, is it doing the same or a different job in placental mammals with different implantation strategies? And so we use cattle and human as two extremes of implantation strategies. Cattle have very superficial implantation, humans have very invasive type of implantation. And what Heidi has been able to identify is actually this protein changes not just the transcriptomic landscape of the endometrium in a species-specific manner, but it also changes the secretions from the endometrium. And that's another key component and key function of endometrial function. And we've actually managed to be able to show as well that there's an effect on attachment of the embryo to the endometrium by these proteins. So that's really interesting. It's a protein coding component of the genome. But the next question we had is what's driving diversity in pregnancy in mammals? And so one of the questions we had when I sat down and talked to a computational molecular evolutionary biologist is, what are the key regulatory networks that had to have evolved in mammals that allow the embryo to signal to the endometrium? And so are they the same signals in all mammalian species, or are there different signals in different species with different implantation strategies? And we chose to look at, in the first instance, microRNAs. And the reason we looked at microRNAs in the first instance was because normally when you get a burst of innovation in evolutionary terms, so when animals formed a front and a back, or you get a placenta formed, there's a coordinate burst of microRNAs that occurs within placental mammals. And so what Ali did, one of the graduate students, is she looked at all of the high quality genomes and asked the question, what microRNAs emerged when placental mammals emerged and haven't been lost since? So she computationally identified this list of microRNAs that arose when placental mammals arose, haven't been lost since. And we then asked the question, are any of these regulated by key molecules involved in early pregnancy? The notion being that if they're involved in implantation, they should be modified by some of those molecules that are important for implantation in different mammals. And so Ali actually identified that when we add conserved molecules to either human or bovine, so these two extreme examples of implantation, we do modify these conserved microRNAs, whereas when we add species-specific molecules, such as the pregnancy recognition signal, we don't identify any changes to these microRNAs. And we've actually managed to also determine that when we change the expression of microRNAs in the lab in vitro, they modify the ability of those epithelial cells to facilitate implantation. And so what we think we've identified here is some of the core components that are required for the signaling between the embryo and the uterus in placental mammals. But one of the things we're really interested in understanding now is if you are an animal that's in a specific clade of placental mammals, and you have this specific microRNA repertoire, are you more or less likely to have an invasive type of placentation? So we were initially focusing on the things that are conserved, so amongst all placental mammals, and now we're really interested in understanding are there signatures of microRNAs that mean you're going to be a very invasive type of implanter, or you have a superficial type of implantation strategy? And what are those types of pathways that are being regulated amongst different mammal species? And so we're also interested in understanding if there's a likelihood that you're going to be more or likely to have a discoid type of placenta or not, depending on the degree of interactions between the maternal and the embryo environment. And so these are really new data, but I think it's nice to be able to show the fact we've gone from looking at a small number of conserved molecules that we think are important amongst placental mammals to being able to expand out and look at diversity in implantation strategies. So this is work that's been ongoing with Mary O'Connell, a member of her lab as well. These are all computationally predicted things, right? And so one of the things we're really keen to do is to be able to test these predictions in the lab. But the uterus is a really complex organ, and it's really difficult to study those interactions very well. And so one of the really important components of what we do in the lab is to try and develop these new in vitro model techniques so we can test all of these wonderful computational predictions and see, actually, do we have a functional effect on implantation? And so when we do our model development, we like to think about different things that make the in vitro system much more like the in vivo system. And so we think about generating structure, flow, and secretion to our model systems in the lab. And so lately we've been trying to develop these extracellular scaffolds where we've tried to lay over luminal epithelium and glandular cells and stromal cells underneath. And we're developing these in a couple of different species, again, to try and be able to test lots of these computational predictions. We're also trying to develop the glandular organoid components to these, and again, building up this really nice in vitro model approach to be able to test lots of our computationally predicted model systems. The last question that we're interested in looking at in the lab at the moment is, how does the environment, so the maternal or wider environment, affect these interactions and endometrial function? And so we've done a lot of work around understanding and developing microfluidic systems to be able to test whether or not changes to maternal glucose or insulin environments alters endometrial function. So for example, here we've developed this kind of dual chamber device where we can change the flow of things like glucose and insulin. This modifies the cellular transcriptome, but also the secretome, and that's what the embryo is actually exposed to. We've done the opposite approach in a human model system where we've looked at overnutrition as an effect. And again, using these model systems to better recapitulate in the lab what we know happens in vivo. More recently, we've been interested in looking at the wider environment. And one thing in particular is understanding heat stress events. I'm having one myself right now. Particularly understanding how if you're an individual that's been gestated in a high heat stress environment, is that going to have an effect on your life course health or life course health of the offspring? And this is some work we've done in collaboration with colleagues at the University of Nottingham in cattle. And we've used here a measure of health is an animal's longevity in a herd. So the healthier an animal is, the longer it's going to be in the herd. The less healthy it is, the more soon it's going to be exited from the herd, is a nice way to put it. And so what we did was investigate if these individuals had a heat stress event in the first 3 months, the second 3 months, or the third 3 months of pregnancy, did that influence how long they stayed in the herd, i.e. how healthy they were going to be over their life course. And what we identified is that if you were gestated in a heat stress event in the first 3 months or the last 3 months, you were less likely to be maintained in a herd, i.e. less likely to be healthy across your life course. And so I think this is something that's a really interesting phenomenon. And this is leading me to the future, and I'm going to stop talking now soon, I promise, but not until I talk a little bit about some of the really important future environmental components that we're interested in looking at. And again, we're here in Brisbane. I don't need to tell you, heat stress is a thing, particularly if your genotype isn't matching to the outside environment. But I think, not to joke, but I think climate change is something that is affecting everybody. It's a really, really important area, I think, in terms of future development for what we do. And I've been really fortunate to be able to get funding with some colleagues from the Wellcome Trust to understand how heat stress in the peri-conception period affects life course health of offspring. Heat stress in terms of if you live in a temperate climate, so probably more Northern European, but if you are exposed to an extreme of temperature of around 35 plus degrees in the peri-conception period, what is the consequence for that for the offspring? So we know already that that does have an effect on pregnancy success and there's higher pregnancy loss. But what actually effect does that have on life course health is something that we have no idea about at the moment. And this brings me to the fact we've been able to put together this, I think, wonderful approach, which is truly interdisciplinary. So we can't test that hypothesis in humans, right, at the moment. But if we use an interdisciplinary approach, like lots of novel large animal model systems, lots of bioengineering approaches, and lots of computational approaches, that allows us to be able to investigate these really important questions and transgenerational questions. And so I think this would not exist without having a diversity of people in reproductive biology and on the fringes, and that can collaborate and interact with one another. And I think for me, one of the really important things going forward for us as an environment of scientists is that we have that diversity across species that we study and approaches that we use, and we're not just looking at one specific thing. So this is something that we've just started, and I think it's going to be a really, really interesting study from a scientific perspective, but hopefully it will have some really important real-world applications. So I think one of the major challenges going forward are going to be climate change effects. One of the other things are looking at diversity of species in terms of conservation and investigation. So I just really like looking at uteri from different species because they're wacky and weird in a wonderful way. And that's a really interesting thing to look at. But also, if we think about trying to conserve species, I think it's really important that we understand not just the oocyte or the sperm or the embryo, but all parts of the reproductive process if we're going to be able to conserve species in the future. And looking at its diversity amongst lots of different types of placental mammals, including the wonderful types of animals you guys have here in Australia. And I just wanted to put a picture of me with an echidna because it was one of the best things that I've ever done. But it really is important that we celebrate the diversity of species and study these in a really scientific and methodical approaches. And I think we can only do that if we collaborate across or discipline as well. Finally, the last thing I wanted to talk about in terms of environment is actually how we interact with one another as scientists. I think this is really important. I think that one of the things, if you don't have your people or your group, form them. We've done that with Repro North, which is a kind of grouping of some random people who do really good repro science and repro science adjacent that are sort of in the north of England and are all people who are wonderful to work with. And we've developed this kind of interactive network where we've begun to publish together, to get grants together, and again just having that environment that's really vibrant with people that you want to work with, right? That's a really, really important component. I think we're really fortunate that we have wonderful scientific communities within reproductive biology, and I think that we need to be better at celebrating how great we all are, and telling lots of other people in other disciplines as well, like why our science is the coolest, because it is, and why they should be involved in it, and really grow our communities, because we have really friendly societies, and I think that's going to be really important going forward. The last environment I'm going to talk about actually is our groups. So for me, something I strive for as a PI is to have an environment in my lab that's really supportive. I think science in general is not shy of people who are smart. Actually, there's probably too many smart people in science, and how they're celebrated and stuff. And I think actually it's really good to reflect on who we celebrate, why we celebrate them, and how we support the people in our labs. Because I think for me, having diverse voices and people from diverse backgrounds is really important. And you want somebody and anyone in your lab to have a wonderful experience, no matter what their lived experience has been, and that they have the chance to do this really cool job in as supportive an environment as possible. And that at least is something I strive for all of the time. I don't always get it right, but I think it's really, really the one environment I think that we can actually, that we have some actionable over. So with that, I'm going to stop talking soon. But the other thing I really want to make clear is I'm here today, but science is absolutely not a solo endeavor whatsoever. And no more today, I think we interact with so many people. And I just want to spend like 2 minutes thanking lots of people, okay? So I want to acknowledge people from my life in UCD, Pat Lonergan, Trudy Fair, and Bea Fernandez, who have been massive supports, both scientifically, but also non-scientifically in my life. And they're people who I don't just go to talk to about science, but they're friends as well. And a massive thanks to them for their support for many, many years. My wonderful Brazilian colleagues, people at Leeds, people like Mary O'Connell, who we do the evolutionary biology work with. And this is some of our Wellcome Trust grouping. Kevin, David, Virginia's not in the picture, and Ramiro as well. My wonderful, wonderful lab and its many iterations, the funders who have supported the work, but particularly the people in my lab. I was an UPI, and I think it does take a bit of a leap of faith for people to want to join your group as an UPI. They don't know what they're getting. You don't know how you're going to do the job, or if you'll be any good at it, high level winging it, etc. But they're a wonderful group of people, and I feel really, really privileged that I get to do this really, really cool job with these awesome, awesome humans. So yeah, massive thanks to them. Finally, I want to say a massive thanks to probably the biggest environmental influence, which is my family, my mum and dad, my brother or sister who's here today, my brother-in-law and sister-in-law, and these 5 wonderful humans that I share some bit of DNA with. They're absolutely awesome, so they are. And with that, Jera, Tá áthas an downer mar tá mé isteach in Brisbane inniu, agus mar atá SRB, SRF agus SSR ag obair le chéile. Tá si go hintach ar fad agus na daoine bí ag éislom ag continue gur míle maith again. Thanks very much. I will stop talking now.
Wow. That was just an amazing presentation, Niamh. You kind of lost me right at that last slide. I didn't follow everything, but I thought I was keeping up pretty well until then. So many important messages, so many questions that come to mind after seeing it. But because of the prestige of this, there aren't going to be any questions. Niamh is going to join us out here for drinks in just a moment, so I'm sure you catch up with her. She's also kindly agreed to stay on for SRB, which will follow after this as well. So you'll be able to catch her over the next 3.5 days. Probably in the bar, I'd guess.
There were some pictures of me not drinking.
At some point anyway. So please see you over that period of time as well. All right. So thank you very much, Niamh. We are really, truly honoured to have you here. Thank you very much for making the long trip out. Thanks, Jean, for coming. Thank you for not actually killing Niamh during her childhood as well. Questions about that as well, but I'll catch up with you about that earlier. But I'm glad you weren't successful. The next part of our proceedings here is a short presentation that Marilyn and Jeff Shaw have prepared to honour, again, Roger's life and Roger's career. We are just getting that presentation loaded up now. And following that presentation, there'll be the awards ceremony in which the medal will be presented to Niamh as well. So,
thanks, Niamh. Thanks, Brett. Is this on? Doesn't sound as if it's on. This presentation is a self-progressing presentation, so I won't be speaking to it. But after the end of it, we'll be presenting the medal to Niamh.
And studying those elephants was just so amazing. It really blew my mind. set the theme for what I want to talk about, which is a plague of people. And my first PowerPoint is you, the audience. I want to use you for a second. Could I have hands up those who have more than one brother or sister? Just look around the room. Wow. I think I could almost sit down. My point is made. But what brought us here, all of us, was that we have been inspired in one way or another by Darwin. Roosevelt delineated the four great freedoms: freedom of religion, freedom of opinion, freedom from hunger, freedom from want. But the women of the world deserve a fifth freedom: freedom from the tyranny of unwanted fertility. I like to say we've discovered a pill that will stop global warming. The oral contraceptive pill. If women used it, human population would come tumbling down, and we would no longer have a problem of too many people. There is a new truth and a new inspiration for me behind that age-old phrase, which is at least 2,000 years old. Touch wood. If we could replant the world's forests, we would do something immediately that would help to redress excessive population growth. Henry VIII was the last person to plant a new forest in Britain. Let's have another one. Thank you.
So, Niamh, congratulations on being the inaugural winner of the R.V. Short Medal and Lecture. It was an interesting exercise, I can say, and I thank Jock Finlay for helping me get this through the various presidents. And I thank Jeff for helping put this presentation together. If Roger was here and he'd heard your lecture, he would have 100 questions. But the first question he would ask you is, Trinity College Dublin, did you investigate E.C. Amoroso's work? Famous placentation biologist, Amo. No?
I wasn't a great undergrad, to be honest.
Anyway, it gives me great pleasure to award you the inaugural Roger Short Lecture and Medal. Congratulations.
Thank you.
Okay, that just leaves me to wrap up proceedings. Thank you very, very much for coming today. Thank you for your attendance and supporting the inaugural R.V. Short Medal and Lecture. It's been a fascinating afternoon, I'm sure you all agree, and we're very grateful again, both for Marilyn and for Niamh, for their attendance here today. Thank you to all that have joined us on Zoom. Although we can't see you, it's been lovely having you present as well. We're going to take a break now, and we have drinks and some nibbles out here in the foyer. And we'll continue until I've been told we need to wrap up about 4 o'clock. Cassie, is that correct? 4 o'clock. Okay. So if you now join me in thanking both Marilyn and Niamh, and then join me out here for some drinks. Thank you very much.